Hey guys, in this video we're going to go over the membrane structure of cells, which is unit 1.3 of the current IB biology syllabus. Okay, before we go over the known structure, as we know is correct today, we're going to go over previous models of membrane structure that are not correct. Anyways, a model is a conceptual representation used to explain and predict phenomena. You guys might need to define this on a test, so if you guys didn't know what a model is, there you go. Anyways, Gorder and Grundle in 1920 proposed the bilayer structure of the phospholipid membrane. How did they come to this solution or to this conclusion? What they did is they extracted phospholipids from the plasma membrane of red blood cells. They laid the phospholipids out in one layer, in a monolayer, and calculated that the area that the phospholipid occupied when arranged in a monolayer was twice as large as the area of the plasma membrane. So therefore, the phospholipids probably arrange themselves in a bilayer. That way, they would occupy the same space as the plasma membrane. The Davison-Dinelli model of 1935 took the findings of Gorder and Grundle and said that the two layers of protein surrounded a phospholipid bilayer like a sandwich. Therefore, this Davison-Dinelli model was also known as the sandwich model. How did they come to this conclusion? Well, they looked at electron micrographs and saw that they showed a railroad track appearance. What I mean by railroad track appearance is, if you see here, that is one cell and that is another cell. Yeah, and that is what I mean by railroad. So basically, that is one cell membrane. That is one layer of a cell membrane, and there's one dark line, one lighter area, and another dark line. And the dark lines, as you see, form somewhat a railroad track appearance. Since the proteins show dark and lipids appear light, Davison-Dinelli said that, well, probably this part is proteins, the dark parts are proteins, and the white parts are phospholipids. And they came up with this model. Well, this model is problematic, and in the 1950s and 60s, some experimental evidence accumulated that did not fit with the Davison-Dinelli model. Those three evidences include the freeze-etched electron micrographs, structure of membrane proteins, and fluorescent antibody taggings of membranes. We're going to go over them one by one. I know it doesn't make sense now, but it will in a while. Freeze-etched electron micrographs involves the rapid freezing of cells, then fracturing them. So they rapidly freeze a cell, fracture them, and they result in something looking like this. So I know this looks pretty disgusting, but all these bumpy things are proteins. Freeze-etched electron micrographs showed that globular proteins are scattered in the membrane, the proteins vary in size, and the proteins don't make a continuous layer like the Davison-Dinelli model suggested. Here's another electron micrograph. This is a nucleus. I labeled it here. And as you see, the nucleus has two membranes, and that is the outer membrane, that's the inner membrane. There are nuclear pores. Some of them have transmembrane proteins in them. That's just another example of an electron micrograph. Anyways, the second evidence that contradicted the Davison-Dinelli model is the structure of membrane proteins. Improvements in biochemical techniques allowed the extractions of protein from membranes. These proteins varied in size, were globular in shape, and were partly hydrophobic. This means that the proteins probably weren't arranged in a bilayer outside of the phospholipids because they wouldn't want to come into because hydrophobic parts wouldn't want to come into contact with the outside environment, outside aqueous environment. Thus, proteins were unlikely to form a layer on the hydrophilic sides of the lipid bilayer. which contradicts the Dason-Danieli model. The last piece of evidence we're going to go over is the fluorescent antibody tagging of membranes. This is basically an experiment by Fry et al. In 1907. What they did is they attached red or green fluorescent markers to antibodies that bind to membrane proteins. The human cell was labeled red, so the antibodies of human cells were labeled red, and the antibodies of mouse cells were labeled green. A human cell and mouse cell was fused together, and the markers, red and green, were initially separated. So let's say if a cell looked like that, the red ones were all, the red antibodies were all here, and the green antibodies were all there. And then within 40 minutes though, the red and green markers were mixed over the cell surface. This suggests fluidity of the cell membrane and showed that proteins move freely through the plasma membrane instead of being fixed. If proteins were arranged in 2 layers, they wouldn't be able to move freely through the plasma membrane. Therefore, this contradicts the Dason-Danieli model. All 3 pieces of evidence that falsified the Dason-Danieli model supported the Singer-Nicolson model, also known as the fluid mosaic model. Okay, now we're going to go over the Singer-Nicolson model. So basically, phospholipid bilayers. Phospholipids form bilayers in water due to the amphipathic properties of phospholipid molecules. That is a phospholipid bilayer. Amphipathic means possessing both hydrophobic and hydrophilic properties. Okay, so phospholipid molecules are amphipathic because they have a hydrophilic phosphate head and a hydrophobic fatty acid tail. Phospholipids are stable fluid structures that form the basis of cell membranes. They are stable because they are arranged, because they have 2 parts. The hydrophobic parts always want to stay on the inside of this bilayer because they do not want to come into contact with the outside environment. Therefore, they always arrange themselves into this structure of the phospholipid bilayer. Anyways, membrane proteins are diverse in terms of structure, position in the membrane, and function. Membrane proteins are embedded in the phospholipid bilayer, and there are multiple types of membrane proteins. So integral proteins are hydrophobic on at least part of their surface, so they are embedded into the bilayer. Peripheral proteins, on the other hand, are hydrophilic. They're either attached to the surface of integral proteins or anchored through a hydrocarbon chain to the membrane structure. I know it's confusing, but there's a diagram later, so make sure you guys just internalize this, and then if you see a diagram and label integral proteins, peripheral proteins, you guys will probably understand better. Anyways, protein content of most plasma membranes is about 50%, but there are exceptions. 75% in inner membranes of mitochondria and chloroplasts, because I guess there are more reactions going around and more proteins needed, and just 18% of the membrane of myelin sheath was served to insulate against signal propagation. Just know that there are exceptions to the 50% protein content, but that is the norm, that is the trend. Functions of membrane proteins. Here, there's another mnemonics, cheap C. C stands for communication, H stands for hormone binding sites, E stands for enzymes, A stands for adhesion, P stands for protein pumps, and C stands for channels. Communication. So these membrane proteins communicate because carbohydrate molecules are attached as labels for identification to these membrane proteins. Membrane proteins are also hormone binding sites. The receptor on the outside of membrane fits the hormone shape. The receptor is a protein, so and the attachment of the hormone to the receptor causes a conformational change, and this sends a message to the cell interior. The membrane proteins are also enzymes. Enzymes catalyze chemical reactions, and they can face the interior or exterior cells, depending on which part of the cell they catalyze reactions in. Adhesion. Membrane proteins also form connections between cells in tissues and organs through adhesion. Okay, membrane proteins are also protein pumps, which pump substances through a membrane by changing shape. Protein pumps are involved in the active transport of particles, which occurs against concentration gradients. I believe that Unit 1.4 is will cover active transport, passive transport, and whatnot. So make sure you guys check out that video if you guys want to know. Anyways, and the last C is channels. So membrane proteins are also serve as channels. That these proteins span the membrane and provide a passageway for particles to move down a concentration gradient, also known as passive diffusion. Okay, so cholesterol in membranes. Membranes also contain cholesterol. It is a component of animal cell membranes. They don't exist in plants. Cholesterols are steroids. A type of lipid. We will go over lipids in Unit 2, in Big Unit 2, and I will post that soon. So if you guys want to check that video out, go ahead. It's hydrophobic for the most part, and one end has a hydroxyl group, which is hydrophilic. It's positioned between phospholipids, and it reduces permeability of the cell membrane. Cholesterol affects the fluidity of the membrane. Basically, in warm temperatures, it reduces fluidity by restricting phospholipid movement. In cool temperatures, it keeps phospholipids from solidifying, increasing the fluidity of the membrane. So, now, just to summarize, just look and see if you guys could identify the structure of each, identify the structures and label them. You guys will probably need to know how to draw the membrane, so make sure you guys know how to draw this whole thing. Anyways, that is a glycoprotein. You guys will need to know what a glycoprotein is. Basically, a glycoprotein is a protein with a sugar group attached to it. As I went over earlier, this sugar group serves as an identification factor. This is an example of a protein that functions as a source of communication. And yeah, that's the carbohydrate train, or the sugar. That is an integral protein. Integral proteins are embedded in the phospholipid bilayer like this. That is a protein channel. Protein channels have this little channel in the middle of them. They're integral proteins with a little split in the middle. They're responsible for diffusion. Peripheral proteins are on the outside of the cellular membrane. They're hydrophilic. And these green structures are cholesterol that I went over. They control the fluidity of the membrane. Make sure you guys know how to draw this whole thing. It is important that you guys understand what each and every component of this structure does, and yeah, and the phospholipid fatty acetyl phosphate head, pretty straightforward. Anyways, thanks for watching. If you guys want more videos, please subscribe and like my channel, and well, thanks. That's it for today.